Jan and Aswan's already joined with us.
Fantastic.
Assalamualaikum Pak Jaim.
Assalamualaikum, bagus.
Bagus.
Halo Julian, How are you?
Bagus.
Baik Padang.
Julian nanya apa itu email Anda itu. Dia minta email Anda.
Iya. Saya nasi Padang.
Di Padang.
Julian udah lama datang ini ke Indonesia, udah sering kelihatannya.
Oh sering.
Sampai kenal nasi Padang segala.
Oh, makanannya makan apa saja.
His favorite food.
Malaysia itu Pak.
Enak.
Mika, saya kok cuma lihat tiga foto ya Pak Zaim, Mika sama Julian, yang lain-lain nggak kelihatan.
Iya betul.
Saya nggak bisa dia.
Terus video saya juga hilang ini. Settings video on off-nya nggak ada.
Saya bisa semua.
Saya nggak bisa.
Kalau attendees nggak bisa.
Oh,
karena
Oke.
Why you don't tell me before?
Sorry about that.
Sorry, beda kelas ya.
VIP dia.
Ini partisipannya, makanya nggak bisa.
Tapi Mika bisa lihat saya, Mika?
Bisa, Pak.
Bisa ya?
Ya.
Udah berapa orang yang hadir, Mik? Banyak.
30, Pak. So far.
Selamat siang, Pak Dekan, Pak Irwan Melanu.
Oh, Pak Irwan mah panelis ya.
Langsung
dihapus.
Hello, Julian. I introduce you the Dr. Irwan Melanu. He's the dean of our faculty.
Selamat siang, Pak Prof. Jaim. Good afternoon, Julian. Very nice to meet you.
Nice to meet you as well.
Well, I need to modify my background for a while. I just want to make sure that the background fits with these webinars.
Okay, probably we can start in 2 minutes or so. So
okay.
Wait for the participants to come in because there are a lot of them. So our technical team is still working on putting the participant in.
Waiting room.
Yeah, in the waiting room. So we will start in 2 minutes or yeah.
So, Mika, you open the waiting rooms open again for the attendee. Right?
Yep, already Pak. Baru dimasukkan satu-satu oleh Nadila dan Rifki, Pak.
Okay, ya. Oke, sudah. Mau dimulai, Mika? Atau gimana?
Sebentar, mungkin ya 1 menit lagi, Pak.
Oke.
Ya. Sambil nunggu partisipan.
Belum pada masuk kok, Mika.
Sudah, Pak, sudah mulai masuk, sudah 50 ya, sudah 50. Jadi mungkin nanti menyusul.
Belum sama di sini, kok Pak Jem nggak bisa lihat? Darumnya.
Bisa sih harusnya. Oh, nggak bisa ya.
Makanya Pak Jaim nggak lihat.
Ya, saya hostnya soalnya.
Oh.
Usah pelit. Emang kenapa sih, Mik, kalau nggak bisa lihat, Mik? Kalau
Apa?
film menghambat ya kalau itunya. Jadi
Oh enggak,
lelet ya.
memang settingnya untuk webinar Pak, bukan untuk ini, bukan untuk meeting. Ya.
Oh, gitu.
Sudah, mungkin kita bisa mulai sekarang karena partisipannya juga sudah mulai muncul, sudah mulai datang. So we will start now. Julian, right?
Okay.
So good afternoon, everyone, or good evening for Julian, and maybe good morning for Hanneke. Yeah? Welcome to our first talk of Paleo Talk series held by Paleontology and Quaternary Geology Research Group, ITB. So with this webinar, we aim to spread our works, our research, and the development in paleontology, especially of what our research group and our collaborators have been done and still doing until now. So I am Mika Rizky Puspaningrum. I'm a paleontologist and I will guide you through this webinar. So before we officially start, I would like to give a huge appreciation for Julian, who amid his very busy schedule was happy to take our over to give a talk and discuss with all of us in our webinar series. Welcome, Julian.
Thank you.
And I will also like to welcome Pak Irwan, our Dean of the Faculty of Earth Science and Technology in ITB, and of course, all of our honorable participants all over Indonesia. And I can also see some from abroad. So we really thank for your participation. So this Paleo Talk series is fully supported by Faculty of Earth Science and Technology, ITB. And thank you for all the facilities that we can use, Pak Irwan. And also by Geological Engineering Study Program. So thank you also Pak Aswan and Pak Yan. And at least we also supported by Ristek BRIN. So our program today will be a short preface by our head of research group, Pak Zaim, followed by our dean, Pak Iran Melano, and the main talk today will be delivered by, all of Julian Lewis. And during our talk today, please mute your microphone. If you have any question, please write it in the chat feature, and please mention your name and institution before. We will read your question or ask you to ask the question directly later after the talk. So without further ado, please welcome Pak Zaim to give a little bit of preface. Please, Pak Zaim.
Baik, terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Ibu dan Bapak sekalian, para peserta dalam webinar ini, Dr. Luis and attendees, on behalf of Kakak Paleontologi dan Geologi Kuarter or Paleontology and Quaternary Geology Research Group from Department of Geology, Faculty of Earth Science and Technology, ITB, we are proudly to say welcome to all of you to attend first Paleo Talk, an online presentation held by Paleontology and Quaternary Geology Research Group in this evening through the Zoom systems. I would like to thank for your participation to attend this online seminar. Dr. Julian, we would like to address our huge appreciation and the deep respect for you to your time to give the presentation in this webinar. This Paleo Talk, first Paleo Talk is an online seminar or webinar program based on our joint research between ITB, Paleontology and Quaternary Geology Research Group, ITB with your university, Griffith University, as a part of our research program in Sumatra. And as we know, we have the collaboration research in Sumatra, in Padang Highland, in Rio area, and the last one in the Bangkok area. Yeah. So you will talk about preliminary result our field work in Bangkok. I think you will talk about this. So this is a very important preliminary research from our joint research, and we have to present that's from the public Indonesia as also as a proof for respect when we asked permit for research in the field last year, I think. So, Dr. Luis, this is online supported by Faculty of Earth Science and Technology, and also Respect Office in Jakarta. And this seminar also link live through the YouTube channel. So the people abroad, everywhere will attend through this Zoom or by the YouTube channel. So it's a very important participation for the public to see our webinar this evening. So I would like to address my acknowledgement for Mika, for Maria, and also Nadila, and of course another committee who work hard and good job for this seminar, and of course for faculty, for my dean, Pak Dr. Irwan Melano, especially also for Vice Dean for Academic Affairs, Dr. Agus Ramdan, who give us in this seminar all facilities, including this Zoom systems there. So with my deep respect for my dean, I would like to invite the dean to give the short speech and open this officially this seminar. So please, Pak Irwan Melano, Dr. Melano, to give your short speech and open officially this webinar. Thank you so much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Pak Zaim. Selamat siang Bapak Ibu sekalian. Assalamualaikum warahmatullahi wabarakatuh. Hello, good afternoon everyone. Friends and students. I'm Irwan Melano, Dean Faculty of Earth Science and Technology ITB. I'm pleased to welcome you to the first Paleo Talk, the Savannah Corridor in Southeast Asia, with special reference to the fossil record of Bangka, Sumatra. This is part of the series of Paleo Talks and discussions related to the field of Earth Science and Technology. Today's talks aim to disseminate the result of research collaborations between ITB and Griffith University in the field of paleontology. I'm sure we are also reaching out to many new friends, especially new friends for me. I'm really glad to meet you, Julians. And also, I believe several students, not only undergraduate, but also graduate students, professionals, researchers, lecturers from other university also join this webinar. A warm welcome to you all. We all understand the importance of Savannah corridors. That may have provided a convenient route for the rapid early dispersal of modern humans through the Southeast Asia into Australasia. So I would like to thank Julian Lewis from Griffith University, who has been working with ITB since, I believe, since 2016. Several papers in a very good journal have been published together with ITB, which is very important for ITB to have our paper published in a very good journal, especially your collaborations with Professor Jaim. And we really hope this kind of collaboration will continue and will open a new collaborations. And I believe by participating in this webinar, we are in the right place and the right time. Together, let us accelerate the exchange of ideas of paleo-related research topic in South Asia. So I wish you all a very successful webinars. Thank you very much.
Thank you very much, Pak Zaim and Pak Irwan, for the preface and welcoming us. So now without further ado, we come to our main show, the talk by Julian Lewis, the Savannah Corridor in Southeast Asia, with special reference to the fossil record of Bangka, Sumatra. But before that, I would like to show a bit of Julian's CV here, although I know everyone already know that Julian is a very great researcher. So he has already a lot of record. So Julian is a senior researcher fellow at Australian Research Centre for Human Evolution, Environmental Futures and Research Institute, Griffith, Australia, Brisbane. Griffith University in Brisbane, Australia. So he graduated from the University of Newcastle for his bachelor degree and from UNSW for honor and PhD. So his research interest here is in vertebrate paleontology, paleoanthropology, evolutionary paleoecology, so archaeology, and so on. And as can you see here, there's a long list of work experience and publication that it will take forever for me to read it, so I won't read it myself. You can just search it in the Google Scholar. So without further ado, please, Julian, have a talk.
Thank you very much, Mika. Thank you, everyone. Thank you, Pazem. It's an absolute pleasure for me to be here tonight, or this afternoon for you, and to give this presentation. So I'm just going to share my screen and hopefully this will work. Okay. Now, can everybody see that? Yes?
Hey.
Yes, you can see.
Okay.
Okay, great. Thank you. All right. Well, I'll make a start. So yes, as has been introduced, I'm going to be talking about the Savannah Corridor in Southeast Asia, and especially want to reference the work that we've been doing in Sumatra and in particular Bangkok, which we went to last year. And as has been said, this is part of a much broader research program looking at paleo environments of Sumatra during the Pleistocene. And this is a collaborative effort between myself and Griffith University and Zaim, Ian Rizal Aswan, Mika Pusparingram, and Agus Trihaskario from Institute of Technology Bandung. And I also want to shout out to Gilbert Price, who's also been accompanying us on many of these field trips in Sumatra. I'm sure I don't need to introduce Sumatra to all of you, but just a quick start. It's the second largest island in Indonesia, and it's actually the sixth largest island worldwide. It's basically a long, narrow island, and it straddles the equator. It is home to numerous large-bodied species, and this is one of the things that actually fascinates me about the island. It's one of the few places on Earth where you can still encounter some of these large-bodied creatures that have become extinct in so many other places. So things like the elephant, the rhinoceros, the tapir, the orangutan, tigers, bears, and so on. So it's still home to a diverse array of creatures today. The first paleontological work in Sumatra was done by this fellow here. It's a guy called Eugene Dubois. And he went to Padang in the 1880s, in the late 1880s, looking for the missing link. And he thought that that Padang was going to be the best place to try to find that missing link. Obviously, he did not find the human evidence that he was looking for in Padang. And after a while, he decided to move to Java, where he eventually found the holotype, the type specimen of Homo erectus. And he changed his ideas throughout time. And by the end of his life, he actually thought that Homo erectus was perhaps maybe not the missing link, but rather something like a gibbon. But anyway, he started off in Sumatra. And as a result of his work in Sumatra, we got the first fossil records for that island. Most of the work that Dubois did in Sumatra were collections from a number of caves in the Padang highlands. And the two caves where he spent most of his time is Lidah Ayah and Sibrambang. And from these caves, he was able to extract a huge amount of fossils. We're talking thousands of fossil teeth, representing a huge array of different animals, including a whole bunch of different monkeys, gibbons, orangutans, bears, elephants, tapirs, rhinos, pigs, all sorts of things. As I said, he did not find any human remains in Lidah Ayah or Sibrambang. And so he thought that all of the fossil specimens that he was getting, because they represented modern species, he actually thought that they represented very recent deposits. So deposits only spanning the last few thousand years at most. Unbeknownst to him, some of the fossils that he thought were orangutan originally, when they were later studied by Dirk Huyer in Leiden, they actually turned out to be Homo sapiens. And so Lida Aya, in addition to all of these mammal species, also has evidence of Homo sapiens within the caves. This is a sketch drawing of the deposits within the Lida Aya cave that we have worked at for a number of years now. And we engaged in a very multifaceted dating program. So one of the things that we tried to do was to date the flowstone or the calcium carbonate caps on top of the main fossil deposit, as you can see here with my arrow. You also have this main breccia bearing layer here. This is where most of the fossils can be seen still today, eroding out of this matrix. And so what we did was date a number of straws, so little stalagmites and stalactites things within this breccia, as well as the breccia itself, using a technique known as OSL dating, optically stimulated luminescence dating. We also managed to get dates on the base flowstone or the base calcium carbonate layer. And essentially an analysis of all the different dates, the top layer, which is the maximum date, and then the layer here, which is where the fossils are, and the minimum age down here, allowed us to calculate using a Bayesian framework model that the deposit ranged in age from between 72 and 62,000 years ago. And as I mentioned, this is the fossil site where Homo sapiens was found. And so this represents the earliest example of Homo sapiens within the entirety of Southeast Asia. So we have Lida Aya situated right about here, and you can see that it's right on the path of movement for possibly, well, for at least Homo sapiens, but possibly other hominins as well. So there's two major exodus of hominin species out of Africa. The first to make it into Asia is Homo erectus. The first to definitively known to make it into Asia is Homo erectus, and you can see that Homo erectus probably came through Africa, through southern Asia and in through to Southeast Asia, down into this area over here. And then likewise, Homo sapiens, probably around about 100,000 years ago or so, also exited Africa. They moved all over the place, but they also moved into this major area known as Southeast Asia. And this is the focus of much recent study, including our own. Southeast Asia during the last 2 million years was a very different place to what it is today. Unlike today, for most of the Pleistocene, the islands of Southeast Asia were actually connected together, and they basically formed a giant landmass known as Sunderland. This is during glacial periods, so during ice ages, when all of the ice was taken up into the ice, into glaciers and things like that. And so what happened was twofold. You have a reduction in the amount of water that's available for evaporation within that area, but you also have movement of the intertropical convergence zone during glacial periods. And the intertropical convergence zone dictates where a lot of the rainfall is going to occur. And so if you have movement of the intertropical convergence zone away from this area here, as well as less water in general because of the landmass that's been exposed, you have a drying out of this continental area. And this drying out of the continental area has been hypothesized to have formed a savanna corridor basically running through the middle of Southeast Asia. You can see that illustrated in yellow here. This is the hypothesized savanna corridor. And there's evidence for savanna-like conditions from all sorts of different environmental records. So things like pollen, things like charcoal, things like isotopes, things like the animal communities themselves, all indicate that there was at least some grasslands occurring in Southeast Asia during some glacial cycles. And these savanna corridors would have been incredibly important for the movement of hominins through these ecosystems. And in particular, probably the first movement of Homo erectus through Southeast Asia occurred as a result of these environments, these grassland environments. Homo erectus was an African species. It came out of Africa, and it was adapted to grassland conditions in Africa. It would have moved out of Africa and followed these grasslands all the way through down into Java. And this is a reconstruction of Homo erectus that you can see here with the Indian rhino, the stegodon. During that time, you also had hyenas living in the area, and you can see some water buffalo there in the background as well. So one of the main ways that we use vertebrate paleontology to try to reconstruct environments in this particular area is by looking at isotopes. And in particular, we look at two main isotopes when we're looking at mammal fossils. The first of these are carbon isotopes. And carbon isotopes generally uptaken by plants in two major ways. So C3 plants, what we call C3 plants, these are things like trees and bushes. They take up the delta 13 or the carbon 13 isotope at much higher densities than C4 plants. C4 plants are things like grasses. And these are due to the different photosynthetic pathways that trees use as opposed to grasses. And so what happens is with C3 plants, trees and bushes, you have very negative delta 13C or carbon 13 ratios, whereas C4 plants, things like grasses, you have much more positive delta 13C values. So because you have such a difference in C3 plants and C4 plants in terms of the carbon isotopes, and because these carbon isotopes are then passed on to the animals that are eating them, you can actually tell what the animal was eating based on their carbon isotope signatures. So C3 feeders, things like elephants, for example, will have very negative delta 13C values, which means that they were predominantly eating bushes and trees and leaves. C4 feeders, things like grazers, things like cows, will be predominantly eating C4 plants, and they'll have much more positive delta 13C values. Oxygen isotopes is a little bit more complicated, but essentially during times of high precipitation, as you can see here, during periods of high precipitation, you have lower delta 18 values. And during periods of less precipitation, so during periods of evaporation, you have higher delta 18O values. So the relative values of the oxygen 18 isotopes gives you an indication of how much precipitation there was going on at a particular place at a particular time. Because most animals get their water from either drinking or from the plants that they eat, this is recorded again in the enamel of the animals. If we look across all of Southeast Asia, we can start to see some patterns in terms of the predominance of tree eaters versus grass eaters through time. So if we look at all of Southeast Asia, and we look at all of the early Pleistocene, you can see that there is most animals are found within this forest ecosystem over here. But there is still quite a few that are found within this grassland ecosystem. By the middle Pleistocene, you see a massive shift towards these open grassland sort of areas. These are the savannas that we've been talking about so far. By the late Pleistocene, you see a shift back towards predominantly forested conditions, and with still some grasslands remaining. And but in particular, during periods like the last glacial maximum, or the LGM, you would have had dominant savanna conditions. And then finally, in the Holocene today, and through to today, you don't see any animals really in Southeast Asia that are inhabiting these grassland or savanna ecosystems. Most animals are found well within the forest ecosystem type. And you can even see that in animals like cervids and bovids. You can see the range of values that are encompassed by these different animals. So for example, here we have the bovids during the early Pleistocene and the cervids during the early Pleistocene. You can see they span everything from savannas up here down to forests down here. By the middle Pleistocene, all the bovids and the cervids are pretty much living only in grassland ecosystems. By the late Pleistocene, they're again inhabiting both grasslands all the way through mixed environments through to forests. And again, in modern times, almost all of the animals that we see are living in forest conditions. One of the other ways that we can look at what sort of environments were present was by looking at the community structure of the animals. And essentially the way that animals use a space means that they can only occur with other animals. And so this co-occurrence of animals is going to be particular for whether the environments are open, mixed, or closed. And so we can look at the actual structure of the communities, what animals are present in a particular time and place, to work out what sort of environments were present as well. And so here, for example, we have a whole bunch of Southeast Asian sites, and you can see that there's quite a few that were within the closed sort of area, quite a few that are in the mixed area, and some that are in the open sort of environments as well. And just to show you where some of those sites that we've been examining in Sumatra sit, Lidah Aya and Sibrabang, sit very much within the closed types of environments. And so we know that the environments in the Padang Highlands during the Pleistocene are almost certainly similar to the conditions that are found there today, i.e. rainforests. So these are the areas that we've been looking at as part of this larger research project with the team. So as I said, in the Padang Highlands, we've been doing most of our research. And these are the 3 cave sites that we've been doing a lot of work in. Lidah Aya, I've already mentioned. The Laosam pit is just outside of Payakumbuh. We've done a bit of work out there. And also Nalau Gulpin is a new site that we discovered, I think in 2015. And we've been working steadily on describing that site, dating it, and getting the fauna out as well. We did a trip out close to Jambi in 2018 to look for some fossil deposits. Unfortunately, we weren't successful in finding any vertebrate fossils. We did find some root casts and things like that, but nothing in the way of what we were looking for. Bangkok fieldwork, I'll be talking about that quite a bit later on in this talk. And finally, this year we were supposed to go out towards the north of Sumatra, out towards Batang Toru, as well as Nias and those sorts of places. Obviously, because of the situation, we've had to postpone that fieldwork into future years, but hopefully we'll be able to get out there, if not next year, then maybe the year after. And so one of the things we've been doing is looking at the isotope values of some of the animals that we've been finding from the caves. And again, you can see these convex hulls are modern animals, so where the modern animals sit in terms of Southeast Asia. So these are all typically rainforest animals. And these yellow triangles are where the fossils that we have gotten from our sites sit. And you can see that they're sitting very much within the rainforest type of ecosystem. And in fact, when you do statistical analyses of whether they're different from modern rainforest species, we find that they're not. So again, this gives us further evidence that the environments within the Padang Highlands in the late Pleistocene were very much dominated by rainforest types of environments. One other thing that we can look at in terms of how the animals are using the landscape is the actual wear that is put onto the teeth as a result of eating different types of foods. And so when animals, and in particular herbivores, masticate the leaves or the grass that they consume, leaves telltale signs on their teeth, little scratches and pits that you can look at under the microscope, and you can actually quantify them. So here you can see, for example, there's some pits down here. There's some very fine scratches. The first researchers in this particular field just counted the number of pits and the number of scratches and related that to whether the animals were eating predominantly grass or predominantly leaves. But we've moved on a little bit from that, and now we can do entire surface scans of teeth and actually quantify it in an automatic way using sophisticated computer software. And you can see here are 4 different scans that we have from 4 different orangutan teeth that we got from some of these caves in the Padang Highlands. And in fact, this paper was just accepted last week in Quaternary International. This is some of the research that we've been doing with the ITB team. And you can see here, some of them have got large pits, lots of scratches. Some have got lots of scratches and not much in the way of pits. Anyway, you can quantify all these and you can look at 2 or 3 major variables. The first is anisotropy. And you can see here, anisotropy relates to how well aligned and the different pits and scratches are. Complexity refers to the nature of those scratches and pits. And heterogeneity looks at the distribution of those different pits and scratches. Now, complexity gives you an indication of how much hard foods the animal is eating. And for things like orangutans, it's particularly relevant about the sorts of fruits that they might be eating. So if they're eating fruits that are very hard or they have hard seeds, that'll show up in terms of the complexity. Anisotropy gives you an indication of how tough the food is. So if animals are eating very soft leaves, for example, that's going to give you a very different signal than if the animal is eating something like bark or very tough old leaves. And so what we did was looked at the orangutan fossils from the Padang Highlands and compared them to both other modern apes. So in other words, the gorilla and the chimpanzee. And we also compared them to macaques in Southeast Asia living in Sumatra today. And we also compared them to each other. Now, the number one thing that we found was that there was no major differences in terms of what the orangutans were eating compared to most other great apes. However, one of the things that we did find, if you look down here, you can see that the proportion of Pongo pygmaeus, that is the Bornean orangutan, the proportion of anisotropic value that has high anisotropic values in Pongo pygmaeus and the Bornean orangutan is much different, significantly different, in fact, from what we find in the Padang Highlands. So this indicates to us that the orangutans from Borneo are eating much tougher foods than the orangutans in Sumatra were in the late Pleistocene. Now, if you look at the other species, the other major species of orangutan, the Sumatran orangutan, you look at the ecological differences in how they eat foods and how they consume resources, it's very different from the Bornean orangutan. So the Bornean orangutan lives in a rainforest that suffers from all orangutans live in forests that have masting events. So masting events is when you have mass fruiting events. But in between these masting events, so in between when there's an abundance of fruit, in Bornean forest, there's not much fruit to go around. And so what most orangutans do is they use fallback foods, so foods that are tougher and not as great as fruit. Whereas in Sumatran rainforests, in between masting events, in between these hyperabundant fruit events, the orangutans eat less desirable fruits, things like figs, but the figs are available all year round. And so you have a massive difference in terms of the diets of Bornean orangutans versus modern Sumatran orangutans. And what we're showing with our study of the microware is that the fossil orangutans from the Padang Highlands are also eating less tough foods than the Bornean ones. So they probably had very similar diets to what Sumatran orangutans have today. And again, this gives us further indications that the environments in the Padang Highlands in the late Pleistocene were very similar to what you find in Sumatra today. Okay, on to Bangkok Island. So Bangkok Island is a pretty unique sort of island in terms of Sumatra. In terms of the zoogeography, so in terms of the animals that are present there, it actually shares slightly more resemblance to the Bornean ecosystems and faunas than it does to the Sumatran ones, even though it's just right next to Sumatra. It's also home to one of these rare types of forests in Sumatra called heath forests. And heath forests are essentially forests that are kind of very scraggly. They've got lots of open areas, they've got lots of grassy sort of sections in them. And this was noted even back when Wallace explored Bangkok Island. And it's still not sure whether this actually represents a real type of forest or whether that type of forest is a relic of changes that have occurred probably because of people altering those forests for thousands of years. And so we're still not sure exactly what kind of forests were present even when Wallace was there, but more importantly, when during the Pleistocene. The thing that interested us about Bangkok was this report from 1884 by a German researcher called Martin. And he basically described the occurrence of modern elephant fossils within Bangkok. And this is a rough translation that I did, but they report about a very complete, very well-preserved upper left molar on Bangkok, a fossil elephant that's almost identical to the Sumatran species living today. Later on, he goes on to talk about the age of the fossil. He thinks that it's probably later than Pleistocene, so he thinks it's probably Holocene. And so this report of a fossil from Bangkok really got us intrigued about the possibility of there being fossil deposits that we could potentially explore and exploit to try to understand what was going on there. And this is the area that they were reported from. This is the southeast corner of the island. And an area called Nibang, and you can see that this light brown sediment type is very much late Pleistocene, Holocene geological coverage. And so we thought this would be the ideal place for us to look for these particular fossils. These fossils had also been reported on the geological maps, but confusingly on the geological maps they were referred to as stegodon, which is a very different animal from elephant. And so one of the other things we wanted to get at the bottom of was, were these elephants or were these stegodons? And so we set out there, and these are the sorts of conditions that we found when we were out in Bangkok. So the first thing that we noticed, and in fact, you could see this flying over, is the fact that a lot of the island is heavily mined. It's one of the primary areas for the production of tin, for the mining of tin. And so when we went out to try to find some original outcrops where we might be able to find some vertebrate fossils, time and time again, we encountered either these lakes, these bright blue lakes that have obviously been mined out. All of this stuff here has been mined. Or you can still see some of the mining in action on Bangkok today. And essentially they're churning up any sediment and trying to get at as much of the tin as possible. And the conditions were quite wet and quite challenging at times, as you can see down here. We were lucky that we did manage to find some very small number of original intact outcrops, and the type of geological formation that we were looking for was the Wrangham Formation. And the Wrangham Formation ranges in age from Pliocene all the way through to Pleistocene. And it's within the top upper gravelly sections of the Wrangham Formation that the elephant fossils have been reported from. And so we did some exploration of some of the Wrangham Formation outcrops that we found. Again, we found some, you can see here, down here, these are root casts. We also found over here, you can see the impression of a tree. So this is a trace fossil. But unfortunately, we weren't able to actually find any of the elephant fossils that we had been told about in any of the deposits coming out in situ. While they're, just as a little aside, we also explored some of the granite caves that were in the region, but they didn't find anything in that either. So, but I thought I'd throw that in just to show you what the granite outcrops look like. However, we were lucky to run into some villagers who told us about some people that had found fossils in several years previously, and in fact, they found fossils from around about here. So you can see that this area is now completely underwater. So obviously, we would never have been able to find the fossils ourselves. But we were able to go over to them, and we were able to talk to them. And they showed us the fossils, and they very kindly donated the fossils for us to study in our research. And what was recovered were a number of different fossils of probably one specimen of elephants. There's probably not more than one individual. So what we have here, we have a tooth. And in fact, there were 4 teeth that were recovered. Here we have part of the spine. Here we have another part of the vertebrae. Here we have this is probably the most complete element that we have. This is part of a humerus. And we also found a whole bunch of other long bone fragments in that area as well. So the main reason why we want to get at the paleo-environmental information of Bangkok is, again, here's the savanna corridor. And you can see that Bangkok sits pretty much smack bang in the middle of where you would think the savanna corridor, or where people have reconstructed the savanna corridor to be. And so we thought if we could get some elephant fossils and date them and get some paleo-environmental information from them, we might be able to get an insight into the sorts of paleo environments that were present in Bangkok in the Pleistocene. And so our approach to this research has been basically 3-pronged. As I said, first of all, is that we've instigated a dating program. So we've submitted some samples for radiocarbon dating, as well as combined electron spin resonance and uranium series dating. We're also subjecting the remains to histological analyses. And finally, we're going to be looking at stable isotope analyses. And that's including serial sampling along the tooth row. We're going to be comparing between specimens, comparing across species, and comparing between species as well. So radiocarbon, we don't have the dates yet. They're still being processed. So I've got nothing to report on the radiocarbon ages. But the electron spin resonance, we do have some results. So for those of you not familiar with electron spin resonance, it essentially works because once an animal dies, its teeth begin to trap radioactive particles. And you can see here, this tooth here, once it's died, it can start to attract and hold on to radioactive particles. So you can think of this like, you can think of the tooth like a bathtub, and the radioactive particles a little bit like water flowing into it. And over time, the tooth will gradually fill up with these radioactive particles. And so by counting how many radioactive particles there are and how quickly the radioactive particles are getting trapped inside the tooth, you can actually work out how much time it has been since the individual died. And obviously there's some potential issues, such as you have to have a clean reset, I guess, tooth to start off with. So in other words, it can't have already had radioactive signals attached to it. You want to make sure that you can calculate if the flow rate changes at any particular point in time. And also you want to be able to see if there's any leaks within the bathtub to see if some of those signals might be getting lost over time. And so there's all sorts of analyses that you can use, or and things, assumptions that you can put into the model in order to try to reconstruct as best as you can the age since the animal was buried. And so we've done this with 2, I believe, teeth of the elephant that we extracted. And so what we did was, you can see here, this is one of the fragmented teeth that we had, and there was a natural split already in the tooth. You can see along here. And so what happened was we extracted some dentine, we extracted some enamel, and we used these to try to calculate the age of the specimen itself. As well as the teeth, we also collected sediment from the outside of the teeth to try to get at the radioactive signals that are penetrating into the tooth and adding to the contribution. Essentially, what you're looking at here is that the dose, the amount of radiation it gets from the tooth itself is quite small. But the amount of radiation that it gets from the sediment that it was found in is quite high. And so most of the radiation that we're calculating is actually from the sediment. Taking all of these things into consideration, some of the preliminary ages that we have from this particular elephant are somewhere in between 16,000 and 25,000 years ago. So this is right in the middle of the last glacial maximum. So right in the period when we would expect Bangkok to be very much dominated by this savanna corridor. Okay, we've also been doing, as I said, a histological examination of this specimen, and this is being done in collaboration with my PhD student, Pauline Basilia, as well as ANU histologist, Justina Miskovich. And essentially what we're looking at is the microstructural anatomy of this particular animal. And we are using paleohistology to reconstruct the life history of this creature. And so the technique that we use is to start off with, we document the samples that we have, and then we do the sample extraction. You can see Pal here trying to get at one of the samples. We use a Dremel drill in the lab. And this is an example, this is not the elephant, but this is an example of the sort of samples that we take. Once we have extracted the sample from the bone, then we embed it into thin sections. And this is going to be done at the Bone Histology Laboratory at the Australian National University. And then once these have been prepared and thin sections have been prepared, we can look at them under the microscope and we can examine the microscopy, and then we can gather the data about the biology of the animal and relate that potentially to the environments that it lived in, and as well as the conditions that it was found in. So these are some of the preliminary results that we have from the teeth. And in particular, one of the things that we were most interested in with the dental histology is to work out just how quickly the enamel was growing. So how quickly the tooth was growing of that particular elephant species. And you can use this equation here. C equals D of sine of I divided by tan of D minus cos of I, to try to work out just how quickly the tooth is growing. And so D refers to the dental prism formation, which, as you can see here, are these little lines down here, and that's a little subset of this much larger area over here. D is the angle between the striae of the rhesus and the enamel dentine junction. You can see that particular measurement here. And finally, I is the angle between the enamel prisms themselves and the enamel dentine junction. So you can see this measurement over here. So calculating these values for the dental sections that we got from the Bangkok elephant, allowed us to calculate that the enamel was growing at a rate of about 6 millimeters a year. And just to show you a comparison, Mammutus columbi, this is the North American mammoth, has a rate that's been calculated at somewhere between 8 to 14 millimeters a year. The mammoth is a much larger animal than Elephas maximus. And over here we have Paleoloxodon cypriotes. So this is a dwarf Paleoloxodon species from the Mediterranean. It's the only other species where this sort of analysis has been done before. And you can see that it's got a rate of between 4.38 and 8.5 millimeters per year. So our specimen from Bangkok Island is definitely probably closer towards the smaller species, the Paleoloxodon cypriotes, rather than the Mammutus columbi. So this suggests that it's probably living a much slightly faster life, I guess, than the mammoth is. And as I said, the histology work is just very much at a preliminary stage. It's very much ongoing. And the next stage is to look at the histology of the long bone fragments. And so one of the things that we will hope to do is to relate the rate of growth in the dental histology segments with that observed in the bone fragments themselves. From that, we should be able to try to get at the life history of the elephants that were living in Bangkok during the Pleistocene. And this is where we're at with these samples themselves. And here's a little video just to show you the sample embedded itself. And so the next stage with these particular bone samples will be to mount them on slides and to create the thin sections, at which point we'll then be able to analyze them. But the majority of our work has been looking at the isotopes, and in particular the isotopes of these teeth. And as I said, we recovered 4 teeth from Bangkok. These are the 4 teeth here. These are specimens number 8, 9, 10, and 11. And we did some serial isotope samplings along the growth margin of the tooth. So essentially every half a centimeter or 5 millimeters, we took an isotope sample and had it analyzed. So this is the first tooth that we had analyzed, number 8. And so just to orientate you, towards the top of the graph here is more forested conditions, and woodland sort of conditions would probably start to occur around about this level here. Also with the oxygen isotopes, anything above this sort of line here is very wet, and below that is just a little bit wet. And so you can see that throughout the growth axis of the tooth, this particular elephant lived very much in forest. There's actually not much variation in the isotopic signature of this particular tooth. It lives predominantly in forests. And it lives in very wet forests, as you can see here from the oxygen isotopes. Tooth number 9 shows a very similar pattern. So again, the elephant is living entirely in the forest, and it's living entirely in wet conditions. The interesting thing to note about this particular specimen is that you can start to see perhaps there might be a little bit of a pattern going. It rises up and plateaus for a while and comes back down again. This is tooth number 10, sorry. And again, you can maybe just see this slight dip, slight convex shape to the forest. But again, this is very small amount of variation, and the animal lives completely within the forest. Likewise with the oxygen isotopes, although there's this jump over here, it's still very much in wet conditions. And this is the last specimen that we looked at, the less samples, but again, along the growth axis, living completely in forest and living completely in very wet sort of environments. So how does this compare to other species and taxa within the elephants? So this graph shows the elephants from Southeast Asia. So these are all the elephant fossils that have been assigned to either Elephas, Hysodriindicus, or Elephas maximus. And what we have, excuse me. So what we have on the top of the y-axis is very dry conditions. At the bottom of the y-axis is very wet conditions. And for the carbon signature, along the bottom right, we have more grassland conditions. And on the left, we have very forested conditions. The first thing to notice is that this species of elephant, Elephas hysodriindicus, is very much living in grass dry sort of environments. So this is what you would expect in savanna sorts of conditions. The Bangkok elephant is found right here. It's closest to late Pleistocene elephants from a site in southern China called Bangxian. So Bangxian is perhaps just very slightly more forested, but just a little bit drier. But these changes are very, very small. They're not significant. Likewise, late Pleistocene Elephas maximus from Java, very similar sort of wet conditions, and again, very much forested. All the Holocene and modern Elephas maximus are found right here. So again, very much forested. Late Pleistocene Elephas maximus from another site in southern China called Kutzai is a little bit drier, but still very much in forested conditions. And finally, Middle Pleistocene Elephas from Indochina, again, is although it's probably the most grassy, dry sort of elephant, it actually isn't dry or grassy at all. It's still living very much in forests and still living very much within much more wet conditions. One of the things we can do, because we have the rate of growth of these particular teeth, we can actually start to work out how the isotopes are changing through time. And this is a comparison that I did with with three teeth that have been serially sampled as well. And these are the site from Baxian Cave. So these are the elephants from Baxian Cave in southern China. And the thing to notice about this is, as I said, Baxian Cave is slightly more, got slightly lower delta 13C, so slightly lower carbon, maybe slightly lower, maybe slightly more forested, but not really. But that's pretty much the only difference between these two. That's obvious from looking at them, just looking at the length from the base. So looking at the length from the base, serially sampled isotopes. But we can do one thing further than that, and we can actually start to look at whether there's any patterns or statistical analyses to work out whether there's anything going on further with these teeth. And one of the things that we can do is something called a spectral analysis. Now, a spectral analysis essentially looks at whether there's any periodicity within the data. So in other words, if there's anything that's happening on a cyclical cycle. And what you'll notice is that there's two red lines here. So this is the this is the significance level at 0.05, and this is the significant level at 0.01. So in other words, if the blue lines here cross the red lines, it means that there's a significant cyclical cycle going on in the isotopes of that teeth. Now, this is the only one where we found such a cycle. This is the elephant specimen from Baxian Cave, and you can see that it has a peak that corresponds to about 90 centimeters in terms of the interval. That's when things start to repeat themselves. And using our calculation of growth rate of 6 millimeters per year, that gives us a cycle of around about every 15 years. So there's something going on here every 15 years that's being recorded with the stable isotopes. This is another specimen from Baxian Cave. You don't see that same signal. And these are the three main teeth from Bangkok. And again, you don't see that signal, but you do see a signal that begins to approach significance at about 16 centimeters. And that's basically every 2.6 years. And you see that in Bangkok 1910 and Bangkok 1909. And these are that's the wave pattern that I mentioned to you before. And so what we can then do is we can then try to fit one of these recurring pattern models to this data. And it turns out that it's highly significant. So what we can do is we can fit the period of 90 centimeters to the isotope data, and it explains quite a large percentage of that particular data. This is for, again, that elephant specimen from Baxian. But we can try to fit that same pattern to the specimen from Bangkok. And again, you get a very strong and nice fit of the model to the data. You also get a very strong and nice fit of the period of 16 centimeters to the Sumatran Bangkok elephant teeth as well. So all of this suggests that there's something going on every 90 centimeters and there's something going on every 16 centimeters. So something going on maybe every 15 years and something going on every 2 or 3 years. And so this data only just came into me today, so it's still very much at a preliminary stage of analysis. But one of the things that affects climate at those sorts of cycles, so there's two things. The first is the change in sunspots. And there's something called an 11-year solar cycle. So every 11 years, these sunspots go through this complete cycle. And they are said to have quite a noticeable effect on global climate and vegetation. And it also has an effect and alignment with El Nino conditions. And El Nino conditions occur basically every 2 to 10 years. So what we might be seeing with some of these elephant teeth and the isotopes from the elephant teeth are these 11-year solar cycles or these El Nino cycles that are actually affecting the vegetation. So even though these animals are found completely within forested conditions, there may be longer term cycles of environmental change that are being recorded by these teeth. But the one thing that's notable is that although Bangkok sits within that proposed savanna corridor, it is coming out as forested. So we may need to redraw those boundaries of the savanna corridor, at least as far as Bangkok is concerned. Okay, and with that, I would like to thank you very much for listening to me and acknowledge all of these funding sources and all of these wonderful people who helped at various stages of the study. Thank you very much.
Right, thanks a lot, Julian. That was very interesting and comprehensive talk about how we tried to reconstruct the savanna corridor farm in Southeast Asia using all the data like histology and isotopic data. And the output itself is very breakthrough because it was the first time we did in, especially in Sumatra. And probably we can be more interesting if I ask the participant to ask themselves. So you can also talk to Julie.
Hosting this, it's wonderful. And it's so wonderful to hear Julian share these results. My question really just was to do with your microware data. Congratulations on the paper, accepted. That sounds very exciting. I just wondered, is it possible that these microware data change with the tooth type in an individual? And or is it more, you're looking more at the dietary regimes per individual or species? And so to that end, what tooth types did you scan for your results?
Yeah, so we've scanned molars and premolars, and we focused just on one particular area of the tooth across all the teeth. And that particular area has been set by people that have studied primates in the past. And so we just essentially, because we're adding to their data set, so we follow the same protocol that they did and focus on the same area of the tooth as they did.
Okay, right, thank you. Thank you.
All right, thanks, Justina, and thanks for joining us. And probably the second question will be from Maudi. Would you like to ask the question yourself, Maudi?
No.
No, probably not. Then I will read the question. So is LIDAR used already in Indonesia for surface mapping? I mean, probably you want to ask if we already use LIDAR for our surface mapping, and
yeah,
I'm
sure I'm not aware of anybody using it, but I'm sure someone is. The thing about LIDAR or LIDAR is it's very good at seeing regular structures. So it's very good at finding old buildings and old, formation, like anthropogenic changes. So when there's straight lines involved or very good circles, LIDAR is very good at finding that. A lot of the work that we're doing is in caves, and caves are natural features on the landscape, and they're going to be very difficult to find even with LIDAR. And you're much more likely to actually find caves using local knowledge oftentimes caves can be, it could look from the top, it can look from just like a normal mountain, but the holes that you enter the caves can be quite small, and there's nothing regular or man-made about them, and so it would be very difficult to pick them up using any sort of remote technology.
Great, thank you, Julian. And the third question will be from Agung Sadayu from UNJ. Please unmute your microphone, Agung. Can you unmute your microphone? Or probably I will just read the question. So your maps say that Belitung is in the center of savanna corridor. Maybe you should also look for the evidence of savanna there. So, and one more question.
Yeah, I've thought about Belitung. The problem with that island is that there's almost no sedimentary outcrops that have been recorded from there. And so the likelihood of finding fossils, it's possible, but it's much less likely than places where sedimentary outcrops have been recorded. And so, yeah, with limited time and money, we have to focus on areas where we know that there's going to be a good chance of finding fossils. And unfortunately, Belitung is not one of those places yet. But if we do hear reports of people finding fossils there, then definitely that would be something that would be interested in chasing up.
Yes, it will be our next destination. And second question from Agus is, West Java were never a part of the savanna corridor as Central and East Java?
Again, the savanna corridor, as I've shown today, has been very much a reconstruction based on various lines of data, and there's a lot of room to move the boundaries of that corridor. It's not, it shouldn't be thought of as these are hard boundaries. And in fact, some places may have been savanna for some period of time and then changed to rainforest, and vice versa. And the thing about these changes is it's not a binary system, so it's not either one or the other. There's usually a transition between those two sorts of systems as well. So you should think of these as very fuzzy boundaries as opposed to very hard boundaries, and fuzzy boundaries that are very roughly put down based on still limited amount of research that's been done on this topic.
Yeah, especially when we have a very mosaic environment.
Yeah.
Even this time, I mean, even this time we also have like a drier area and also wetter area, even in some.
Exactly, and it's all about scale. Which scale?
Yeah, exactly.
Yeah.
That was great. Okay, so we also have a few questions from the YouTube channel. First, from the YouTube, I will read it for you. So there are quite a lot of questions here. So the first question will be from Wildan Nusantara. So he's also one of the micropaleontologists. So is there any fossil from Sumatra? This is a very, very great question, I think. And why is the closest is Baxian? Any correlation with the tectonic? Okay, that's probably a bit of outreach, but yeah.
Sorry, so could you just repeat the question?
Okay, so is there any fossils from Sumatra? That's
any fossils?
Yes.
Well, yes, fossils, yes.
What kind of fossils?
Sorry?
Yeah, so
yeah, I mean, yes, there are many fossils. There's vertebrate fossils, there's microfossils, there's invertebrate fossils. There are lots of fossils from Sumatra. Yes, absolutely.
Yeah, exactly. Probably what he's trying to ask is more of the vertebrate fossils that we find in Sumatra, because obviously compared to Java, the vertebrate fossils in Sumatra is, let's say, doesn't have any longer age or older age.
It actually does. So the fossils, yeah, that's true. So there are no early Pleistocene vertebrate fossils that I know of in Sumatra, that I know of at least. And middle Pleistocene, maybe some of the fossils that we found from Nalaugupin are middle Pleistocene, right at the end of the middle Pleistocene, but they're most likely to be late Pleistocene. And there's certainly no extinct species from Sumatra, or not as many extinct species from Sumatra as there are from Java. But we have recovered the first evidence of hexaprotodon from Sumatra. And so I think with more research and more field work in Sumatra, we will find deposits similar to those in Java.
Yep,
but so far we just haven't found them.
He also asked about the elephant fossils in Sumatra. Probably you have already mentioned it, that the elephant fossil in Sumatra that we've found are the modern elephant.
The modern, that's right. So we've got elephant fossils from all of the Padang caves, and that's all Elephas maximus. And we also have the Elephas maximus fossils from Bangkok. There's no stegodons that have been reported from Sumatra as far as I know, and no other proboscideans either.
Yeah. So the next question will be from Benjamin Pereira, our master student. So is there any possibility in the future to take the fossils from Java seabed? Because it was, it was the southern corridor. So considering the huge area of Sunderland savanna during Pleistocene.
That's a very good question. I think it's a very good question, and I think it really depends on the place, the places on the flooded Sunda shelf. So I think, for example, in between Bangkok and Sumatra, there'd be no point dredging for fossils there because all the marine sediment has already been dredged for mining. But certainly in some of the areas further away from the mining operations, you may be able to do some dredging, and you may be able to get some fossils that way. Another intriguing possibility is if you have a high enough or quick enough rate of sea level change, you may actually be able to get some sea caves. And so there may be fossils preserved in some underwater caves in that sort of area as well. And so all of those things are definitely worth doing. The first thing you would want to do, I think, is to just do a survey, a remote survey of the sea floor to work out some of the best places to look. So yeah.
Probably I can help with that because one of our attendees here, our participant here, is Gatot from ITERA. He did a underwater, what was that? The diving, and he found a small cave, and then he found a fossil in the Java Sea. So probably that will be one of the
definitely
to do that later.
Yeah. That's very exciting. It's very interesting.
Good. Yeah. Okay. So the next question will be from Slamet Supriyadi, and he asked about how did the climate when during the Pleistocene time in Bangkok Island. So probably you have already
Yeah. So yeah, it was from the records that we have, it was all forested.
Yeah.
Wet and forested, so very different to what it was. Probably the same sort of precipitation. Well, probably wetter than today and a lot more forested than today.
Yeah. Of course, today is way drier.
Yeah.
Especially because of the mining. Okay, and next question is from Fitraya Ramadhani from detik.com online media. So can we compare the savanna corridor in the Pleistocene era with those we have today? Let's say savanna at Balora National Park in Java, in East Java.
Yeah, I think it hasn't been done. I think that's definitely something that's very much worth doing, very much. One thing that we don't have a very good understanding of at all is there's some small pockets of naturally occurring grasslands throughout Indonesia. But one of the things we don't understand at all is the sorts of environments that they represent and the sorts of animals that are found there, and how those environmental signals might be preserved in the animals that are found in and around those areas. So I think that's a very rich and important area of study in this particular field. Yes.
And then the last question is from Mahirta from Gajah Mada University, if I'm not wrong. So how many meters below the surface that we were found find fossils?
It really depends on the site. So with some of the caves, for example, we've traveled tens of meters down underground to try to find the fossils. And sometimes the fossils are on the surface and we can just see them lying on the ground. And so it just really depends on the site and how much weathering has gone on and the type of depositional environment it is. So all those things come into consideration.
Yeah. All right, that was probably the last question from our session today. So that was a very interesting and comprehensive talk, Julian. And actually, even though I included in the project, I learned a lot about today, especially about the paleoclimate and also how, yeah, the pongo and the elephant is a very interesting data. So yeah, that was great. And we can learn that the savanna is actually not pretty, 1 or 0, it's going to be savanna or the forest, but sometimes it change between or in the middle between the savanna and the rainforest. So that was a great talk. And yeah, thanks for joining us. And
my pleasure.
The participant who would like to get a certificate, please see the chat features. So I hope you can fill in the forms. And for our next talk will be next week on the 31st of August. So please stay tuned. Our guest lecturer or guest will be Gilbert Price. So we're looking forward to have him here. And I think that's it. Pak Zaim, do you want to say something before we close the?
Yes, just a short thank you for all participants in this webinar this evening, and especially for Julian. Thank you so much, Julian, for your time to give talks this evening. And of course, for our collaboration between ITB and Griffith will be fruitful with our research program and publications in the development of research in Indonesia, of course, during our collaboration between ITB and the Griffith. So we have to stay tuned for every time to make a collaboration in the research for development of scientific in Indonesia, of course, and in Australia. So I think that's all for my comment. And again, thank you so much for your time, Julian, and everybody, attendees of this webinar. Thank you so much. I appreciate your time to participate in this seminar. Thank you for all. Thank you, Mika.
And thanks for the breath of fresh air here, Julian.
Thank you very much. It was an absolute pleasure. It was great to see you all.
Yeah, it was a very great pleasure. Yeah. So I will officially end the webinar here and stay tuned for the next Paleo Talk series for next week. And bye. See you all.
Bye.
Thank you.
Before that, probably we can take a picture.
Stay safe, everybody.
Thank you.
Okay.
Yeah. I don't know if we can take a picture, but
okay. Yes. I forgot.
Yeah.
Can you take a picture for us?
Oh, probably not because
Oh, okay.
this we have.
Only for 3%.
But probably Dila bisa yang bapak-bapak yang.
Okay.
So please stay for a bit so we can have
And please show your face, just not for the names.
Okay, great. Thanks, Julian.
Thanks, Julian.
All right.
Next time. Terima kasih untuk Ibu dan Bapak semuanya yang telah bergabung dengan webinar pertama dari grup kami, paleontologi dan kakak paleontologi dan geologi kuarter di DB, dan semoga ini menjadi pemicu untuk kita untuk tetap melakukan penelitian.
Udah pergi.
Ya, terima kasih Mika, terima kasih Nadila, dan terima kasih juga Maria dan semua panitia. Terima kasih.
Ya, Juliannya udah keluar, Pak.
Ini foto.
Bagus.
Juliannya tidur.
Bagus.
Malam kan.
Tambah 3 jam.
Terima kasih Bu Mairta.
Ada Bu Mairta.
Bu Mairta, wah, makasih Mbak.
Terima kasih undangannya.
Terima kasih. Jangan lupa untuk minggu akan datang ini dengan Gilbert yang satu tim masih dengan kami. Kita akan lihat sama-sama nanti minggu depan tanggal 31 Agustus. Ya. Baik, ya sekali lagi terima kasih juga untuk fakultas yang telah memfasilitasi Zoom ini. Pak Irwan, terima kasih waktunya, dan terima kasih juga untuk Pak Dekan, Wakil Dekan Pak Agus Ramdan. Ya. Semoga tidak menutup lagi untuk fasilitas yang akan datang.
Baik, kalau sudah kami akan menutup room-nya.
Ini difoto dulu.
Udah tadi,
udah pergi.
udah tadi Mas.
Oke.
Oke.
Ya, terima kasih sudah hadir
kasih
dalam
juga.
acara ini. Tadi sayang Juliannya malah udah pergi duluan.
Dia udah. Ya, terima kasih untuk semuanya. Ya, assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Wabarakatuh.